In order for a cell to be successful and to reproduce, it has to go through three main steps. The genetic information contained within that cell, whether it's prokaryotic or eukaryotic, has to be copied, it then has to be separated, and the cell has to divide. So with prokaryotic cells, our DNA is going to be not housed within a nucleus. It's just going to be free floating within the cellular structure because it's a simple structure. And it's going to replicate. And that replicated DNA is going to be all bundled up together. But it's going to have two origin, or each strand of DNA is going to have one origin of replication. So when you have two copies of the DNA, then you're going to have two origins of replication. Those origins of replication are then going to begin to segregate to opposite sides of the cell. And there's proteins that are helping them do this, like you see those purple little dot things down in that last structure. And those origins of replication are going to help the bands of DNA separate from one another and form two new cells. So that cell division is where cytokinesis is going to take place, and the cytoplasm is actually going to divide, and a cell wall or a plasma membrane is going to be formed around the both the existing cell and the new cell. Prokaryotic structures really are very, very small, therefore they can reproduce very quickly, like within 20 minutes under optimal conditions. So, for example, with E. coli, when you're cooking hamburger or pork products such as sausage, where you have lots of surface area exposure to the meat product itself, we always recommend that you cook your meat at least 160 degrees. And the reason for this is because if you only cook it at 120 degrees, that bacteria, that is the best environment for bacteria to grow, is at 120 degrees. So it replicates within 20 minutes in those type of conditions. However, if you heat it up to 160 degrees, that is too hot for that bacteria to survive. So it's not going to replicate at all after it gets to about 140 to 160 degrees. So a single bacteria has the ability to produce 1 billion different descendants in just 10 hours. So you have to be very, very careful with bacteria-borne pathogens. So here we look at an electron micrograph of a prokaryotic cell in the division process. So we have our cell wall along the edge here, and you can see that it's starting to divide. It's going through cytokinesis, and then the white structure in the center, this is actually the DNA that is separated and is moved towards opposite poles of the cell. Eukaryotic cell production is much more complicated because more than one chromosome is involved. So eukaryotic cells are going to be diploid, meaning that they have two sets of chromosomes. So if you think about this in terms of your own individual chromosomes, for chromosome 1, you're getting one from mom and one from dad, and they're coming together to form a pair. So your set of genomic information will be in a diploid configuration. Eukaryotic cells, because they're diploid and have two sets of chromosomes, that means that they're going to have homologous pairs. And those pairs are going to be like in structure. They're going to carry the same genetic information with the exception of sex cells. And one of those chromosomes, if we look at this chromosome right here, by itself is considered a haploid because it's only one set of a chromosome. So gametes are going to be haploid. And gametes are going to be important when we start talking about meiosis and eukaryotic cell reproduction, because we're going to take a diploid structure and actually reduce it down through division. I know that sounds odd, but just stick with me. Through the eukaryotic cell process. And so we're going to take a diploid and reduce it down to a haploid. All right, so this is a really good point for you guys to review what we've been over in part A and so far in part B of this chapter. So these are a couple of good questions for you guys to pause and make sure you have a good handle on these definitions and the 3 steps that each cell has to go through, because it's really the central dogma behind cellular reproduction. Really briefly, we're going to look at chromosomal anatomy. A chromosome is going to be the structure that is formed when DNA winds itself up into a double helix. So this is what forms those X structures that we associate with chromosomes and the Y structures. So centrosomes are made up of 3 major components. The first is a centromere, and the centromere is going to be the center part of the centrosome, like you see here, these dark gray areas, and these are where spindle microtubules are going to attach. So spindle microtubules are going to be associated with the cell and the nucleus specifically, and those microtubules are going to attach to that centromere for the division process. The second major anatomical structure with chromosomes is a kinetochore, and kinetochores are proteins that move the chromosome along the microtubules, and they're associated with the centromere. So the green area here is a kinetochore. It's associated with the dark gray area, which is the centromere, and it is going to be a protein that is going to help the spindle fibers and help that chromosome move along those spindle fibers. And finally, the third and final structure associated with a chromosome is a telomere or a telomere. It's pronounced both ways. And these telomeres are the natural endpoints of the chromosome. And they are associated with the end of the chromosome to help protect that DNA at the end of those chromosomes and provide stability. Now, we have some current research, as well as some research done in the late 90s, that indicates that telomeres are actually associated with aging. So if you have really good, strong, healthy telomeres, you may not show signs of aging compared to a person that has weaker telomeres. So finally, as we look at centromeres and chromosomes, those chromosomes are going to be have 4 major different configurations, and it all has to do with the location of the centromere. So you could have a centromere that is directly in the center, attaching chromosome 1 to chromosome 2, right at the halfway point, and that is going to be termed metacentric. You could have a centromere that is going to be attached a little closer to one of the ends. That's called submetacentric. Acrocentric is where that centrosome is going to be very, very closely attached to one end. And then telocentric is where those chromosomes are actually going to be attached to the telomeres. So these are the 4 major structures that centromeres can actually occur in. All right, that is the end of part B for this online lecture. And next we're going to get into meiosis and mitosis and looking at specific cellular reproduction.